Can somebody confirm you can hear me? Can anyone hear me? Maybe my speaker isn't working. Says it is. Turn up the volume.
I can hear you.
Thank you.
Yeah, sorry, I was away from the computer.
It's not a problem. All right. Are there any questions before we get started? Let me shut down a few things. There we go. Don't need that. Let me share my screen here. All right. Hearing anything, so let's begin. Hopefully you can see my screen now. Today is March 5th. And we're supposed to be finishing chapter 13, viruses, viroids, and prions. I'm not sure we'll make it, but we might get close. And let me remind you, your unknown notes pages are going to be closing this Friday, March 6th. So the last day you can request a test is March 6th. And if you request a test on March 6th, which is Friday, by the way, it must be a test for only one day because your unknown notes, or not your unknown notes page, closes on March 6th, but your unknown report has to be turned in on March 7th, 11:59 p.m. March 7th. Any question about any of that? All right, we are on our second to last chapter, so let's begin chapter 13. I mean, chapter 14. Is that right? No, it's chapter 13. Sorry. I'm in the wrong lesson there. All right, I have that we were on this slide and we mentioned that viruses multiply by hijacking a host cell and then using the host cell to replicate the virus. And almost everything the virus needs, except for maybe a few enzymes and then the genes encoded by the virus to make the protein coat, everything that the virus uses to replicate come from the host cell. Very few things actually come from the virus. Let's talk a little bit about viral multiplication in bacteriophages. Let me shut the door here because I'm getting some noise outside. I don't know if you guys could hear that, but I could hear it. Let's talk about viral multiplication in bacteriophages. Bacteriophages are viruses that infect bacteria cells. So bacteriophages just mean viruses that infect bacteria cell. Bacteriophages use two mechanisms for reproducing. There's the lytic life cycle and the lysogenic life cycle. The lytic life cycle, the host cell will be lysed. When the cell replicates the viruses, the last thing the virus does is it lyses the host cell, and then that releases the newly replicated viruses. In the lysogenic life cycle, the host cell remains alive. So the two remain in symbiosis as long as the lysogenic life cycle continues. Any question about the two ways that bacteriophages reproduce? All right, when we're talking about viral multiplication, especially in bacteriophages, what do bacteriophages need to reproduce? What components does a virus need to make?
An energy source.
Well, yeah, it needs an energy source, but I'm asking what it doesn't make the energy source. What do the virus need to make to replicate the virus?
DNA.
Yeah, it needs to replicate its genome. DNA if it's a DNA virus. It's actually RNA if it's an RNA genome for the virus. Remember that viruses can have RNA for their genome, certain species do, and then other species have DNA for their genome. And what else does a virus need to replicate the virus?
Means to enter the host cell, so an appropriate connection receptor.
Yes, what I'm trying to get at, it needs to replicate at a minimum its genome and the protein coat that the virus is surrounded by. Now, obviously, if there's a viral envelope, that would have to be replicated as well. But bacteriophages, bacterial viruses, they need to replicate their genome. And for a double-stranded DNA virus, it will just separate the two strands and then use each strand as a template to make uh, another strand, meaning the complementary strand. Those two strands will come together to make the double strand DNA of the virus. And then it has to get together with the capsid proteins. How that's made is the double strand DNA of the virus separates into single strands, and it'll be transcribed into viral messenger RNA, which will then be made into the capsid proteins. And then the capsid protein has to get together with the replicated genome to make the new progeny virions. Any question about any of that? And that's only discussing it for a virus that's double strand DNA. It'd be slightly different if it's a single strand DNA virus, and it would be different if it was a virus having RNA. And we'll talk a little bit about those differences in a little bit, but not right now. Any question about any of that? All right, let's talk about the viral multiplication cycle of the lytic life cycle of a bacteriophage. There are 5 stages of the lytic life cycle. There's attachment, penetration, biosynthesis, maturation, and then the last stage, release. Let's watch a little video on the lytic life cycle.
First, the phage particles stick to the surface of the bacterial cell.
Shut that off. Sorry, I don't have the program to play a movie, and I need to get this converted into, what is it, a modern thing here. So let me find.
Did you just look something up on YouTube to show us?
Yeah, you could do that if you had it. Oh, what is the thing? I used to know the name of it. There's something V something, but I've got another vid here I can show you. See, that's a movie, and this computer just can't play a movie. So let's go with this one, and we'll only watch the first of it.
In a way, the virus is a parasite.
We'll watch the later part in just a minute. Let me blow that up. There. Oh, that didn't seem to...
It attacks a host cell.
That's not now working. Let me try that again.
In a way, the virus is a parasite. It attacks a host cell and takes over it.
I might not be able to blow this up. I don't know what's going on here. Let's try it this way.
In a way, the virus is a parasite.
All right, I'm not able to blow this up. I don't know what's going on here. So let's just watch a small one. This used to work. I'm not sure what's going on.
In a way, the virus is a parasite. It attacks a host cell and takes over its chemical machinery for making proteins and nucleic acids. One of the better understood virus life cycles is phage, a virus which attacks bacteria cells. The phage begins its assault by attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall, and the nucleic acid from this capsule is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the new phages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle.
All right, let's stop right there, back up just a little bit, and I'll keep this here. And then we'll play the rest of the movie for the lysogenic life cycle. All right, any questions about that? So the first stage is attachment, where the phage actually attaches to the host cell. The second stage is penetration, where you saw it in the movie, a little bit of the cell wall is dissolved right here, but actually it happens because the virus produces the enzyme lysozyme right here only. And then that makes a hole in the cell wall. And then the virus literally contracts where the tail right here contracts, which you can see here, but it puts pressure on the head, and that will inject the DNA, which is inside the head, into the host cell, which we saw in the mid-movie right back. Oh, shoot, it's not going to show me a picture.
Injected through the hollow tail into the cell. And the nucleic acid from this capsule is injected
where the tail contracts, and that pushes the genome in the head into the cell. Any questions about any of that? Let's go back to where we were.
New virus particles can be reproduced in a
about right there. So that's penetration, where the virus actually injects its genome into the host cell. This part here, the protein, just stays on the cell, and it's sort of inactive, immobile, and this virus is attached to the outside of the cell, but it doesn't do anything after it injects the genome into the host cell. Once that happens, the genome, the first thing it does is it will start transcribing the viral genome, and the first gene that it transcribes actually is an enzyme which degrades the host cell chromosome. Why does the virus want to do that? Because the virus wants to call the shots in the cell. And an easy way of calling the shots in the cell is if you degrade the host cell chromosome. And then your genome can call all the shots for what's going to happen in this cell. Any question about any of that? All right, and then we enter the biosynthesis or maturation phase. Biosynthesis is the production of the phage DNA and the phage proteins. How that happens is the two strands of DNA will be separated, and each strand will be used as a template to make the corollary strand, and that can replicate the DNA. Now, obviously, if you're replicating another genome, like single-strand DNA or RNA, it's going to be a little different, but we'll talk about that later. We're not going to talk about it here. But my point is the biosynthesis happens, and that's how the genome will get replicated, and then the genome will be transcribed. And mostly what those genes will do is they will encode for the proteins of the capsid protein code of the virus. And so the virus proteins will be made. And then the virus proteins self-assemble into the virus. And that'll be viral maturation, probably. And so the virus proteins will come together, the head, the tail, the tail fibers. And there's an enzyme that is encoded by the virus, which actually stuffs the genome of the virus, meaning the replicated viral genome, into the head of the virus. And then the virus self-assembles. And then the last thing the viral genome does, meaning the last gene that's transcribed, is the gene for the cell to lyse. And what that is the enzyme lysozyme is expressed from the viral genome, and then a bunch of lysozyme will be made, and it will degrade the cell wall of the virus. Without the cell wall, water can move into the cell, and then the cell membrane will expand, and normally that's held intact by the cell wall, but the cell wall is now degraded by the lysozyme, and so the cell will eventually burst. And then that will release the newly synthesized new virions. Any question about any of that? All right, two terms that you need to know for the viral lytic life cycle are the burst time. That's the time from initial infection of the host cell by the virus till the time when the cell bursts. This averages 20 to 40 minutes, and obviously it will depend on the species of the virus, as well as the temperature you're growing, I guess we'll call it the virus at. Any question about that? And then there's another term you need to know. That's the burst size, the number of newly synthesized virions which are released when the cell bursts. It depends mostly on the species of virus, but usually somewhere between 50 and 200 viruses. Any question about any of that? All right, that's the virolytic life cycle. Let's talk about the viral lysogenic life cycle. During the viral lysogenic life cycle, the virus and the host cell live in symbiosis, and this will continue for as long as the lysogenic life cycle lasts. When a lysogenic virus first infects a host cell, it will inject its genome into the host cell. And we're going to talk about this genome being DNA. If the virus decides to enter the lytic life cycle, the viral genome will be replicated. The viral protein capsids, proteins will be replicated or synthesized, I guess I should say. The virus will self-assemble, new virions will be made, and then the cell will be lysed, releasing the newly replicated virions. So a lysogenic virus can enter the lytic life cycle, and it just proceeds the same as the lytic life cycle we've already discussed. Any questions about any of that? So a lysogenic virus, once it first enters the cell, it has to decide, do I want to enter the lytic life cycle or do I want to enter the lysogenic life cycle? If the virus decides to enter the lysogenic life cycle, the viral genome will actually integrate into the host cell's chromosome. And this is called a prophage, where the virus, a bacteriophage virus, is integrated into the host cell's chromosome. So that's what prophage means. While the virus is in the lysogenic life cycle, the only time the virus can replicate is when the host cell's DNA replicates, because when it replicates, it will replicate the prophage. And then this cell here will get separated into two daughter cells. Each will get a replica of the host chromosome and a replica of the prophage. And that can continue for many cell divisions, where the virus remains in the lysogenic life cycle, replicating only when the host cell's DNA replicates. But every once in a while, for reasons that are largely still not understood, at least well, by biologists, the virus, which was in the prophage, meaning integrated into the host cell's chromosome, can leave the host cell's chromosome. We call that excised. And then the virus enters the lytic life cycle. So the virus can leave the lysogenic life cycle, and if it does, it will enter the lytic life cycle. And as I stated, they've studied this now for over 100 years, and they still do not understand why the virus leaves the lysogenic life cycle and enters the lytic life cycle. There is one event that does tend to switch the lysogenic virus into the lytic life cycle, and that is if you damage the host cell's DNA, the virus can recognize that damage, and it realizes that the host cell may die because its DNA is damaged, and then the lysogenic virus will switch to the lytic life cycle, and then it can replicate. Any questions about any of that? All right, let's watch a little movie on the lysogenic life cycle.
Interestingly, the lytic cycle does not always happen immediately. Sometimes, rather than producing virions, phage nucleic acid incorporates in the host cell DNA.
And remember, that only happens with a lysogenic virus. A lytic virus can only enter the lytic life cycle, but a lysogenic virus can enter either the lytic life cycle or the lysogenic life cycle. And it has to decide when it first enters the host cell which life cycle it's going to follow. And now let's follow the lysogenic life cycle.
Now, each time the cell divides, the phage DNA is also reproduced and is spread to more and more cells. Then suddenly, for reasons that are still unclear, the phage DNA activates and the destructive lytic cycle begins. Once again, the host cell is destroyed, and a new generation of phage are released to invade new hosts.
All right, everything about this movie is correct, except...
And the destructive lytic cycle begins. The host cell is destroyed.
There, they're showing you bacteriophage T4, and bacteriophage T4 only enters the lytic life cycle. It does not enter the lysogenic life cycle. But other than that, this movie is totally correct. What they should have done was draw lambda instead of bacteriophage T4, because that virus can actually enter both the lytic life cycle and the lysogenic life cycle. Any questions about the movie? All right. Let's see. There is a repressor protein, which is a gene on a lysogenic virus in its genome, And the repressor protein keeps the lytic life cycle dormant. So the repressor protein shuts down the gene expression of the lytic or the lytic life cycle genes, meaning the proteins that make the capsid protein of the virus. And as long as that repressor protein is active, the virus will stay in the lysogenic life cycle. I already talked about the prophage DNA is when the viral DNA incorporates into the host cell chromosome. And we talked about the lysogenic virus can switch to the lytic life cycle. If it does, the prophage is excised from the chromosome. And let me just state that after even 100 years of studying this, biologists do not quite understand why the lysogenic virus can enter the lytic life cycle. Why does it choose to switch? All right, any questions about lysogeny, the lysogenic life cycle, or the lytic life cycle? If not, let's move on to the consequences of lysogeny. First, if there is a host cell, that has a lysogenic virus in it, this cell is now immune to infection by the same species of virus. So once the virus gets infected with a lysogenic virus, it enters the lysogenic life cycle. This cell is now immune to infection by the same species of virus. Another consequence of lysogeny is this host cell gains new DNA, and that's the DNA of the virus. That can change the phenotype of this cell because it can express the viral DNA, giving this cell new phenotypes. The best known example of that is Corynebacterium diphtherium. This bacteria only produces the Corynebacterium diphtherium toxin if it is infected with a lysogenic virus, meaning it's the lysogenic virus that actually encodes the Corynebacterium diphtherium toxin. So yes, whether or how sick you will become by a viral infection may depend on whether the bacteria that's infecting you is also infected with the virus. And you would much rather be infected by Corynebacterium diphtherium that cannot produce the toxin than you would by a bacteria that actually had the lysogenic virus that encoded the Corynebacterium diphtherium toxin. Any question about any of that? All right, that's enough about bacteriophages, viruses that attack bacteria cells. Let's talk a little bit about the multiplication of animal viruses. These are viruses which can infect a human cell, meaning bacteria viruses do not infect anything but bacteria cells, and usually only just a species or a few species of bacteria. Animal viruses follow the same basic pattern of viral multiplication, but there are some differences. There are some differences in attachment of the virus. For example, animal viruses only need to attach to the cell membrane. There is no cell wall the animal virus needs to get through. The penetration is also different. It's either by endocytosis or fusion. We'll talk a little bit about that. The encoding could be a little different. Biosynthesis is largely the same. Maturation is largely the same, but release can be different. There are some animal viruses that do rupture the animal cells. So we can call them a lytic animal virus. But there are other viruses that do not follow the lytic life cycle. Most of them follow the budding life cycle. If they are a budding virus, they have an envelope. If they are a lytic animal virus, then they follow the lytic life cycle, or there is one other life cycle we'll talk about. It's very rare, but there are actually 4 animal life cycles. The 2 main ones are the lytic life cycle and the budding life cycle. Any question about any of that? And we'll later talk about the life cycle of animal viruses. Multiplication of animal viruses follows the same pattern of bacteriophage multiplication, but there are several differences. The main difference is an animal virus can replicate either in the nucleus or in the cytoplasm, but a bacteriophage virus can only replicate in the cytoplasm. And why is that? Why can bacteriophages not replicate in the nucleus? Come on, somebody should know the answer to that one.
Wait, there is no nucleus in our prokaryote. I'm confused.
Yeah, there's no nucleus in a bacteria cell, so that's why bacteriophages cannot replicate in the nucleus. Only animal viruses, or maybe a plant virus, we'll talk about them at the end of this lesson, can only replicate in the nucleus. And then a bacteriophage has to replicate in the cytoplasm because there's no place else in the bacteria cell. Okay? So that's the main difference between the multiplication of animal virus and bacteria viruses. And then it actually something else going on about an animal virus. If it can replicate in the nucleus, it's likely to be a DNA virus. And if it replicates in the cytoplasm, it's likely to be a RNA virus. And we'll talk a little bit about that later. So let's talk about multiplication of animal viruses. The attachment will differ, and that is you can't attach to the cell wall because animal cells don't have a cell wall. So the virus must attach to the cell membrane. And then it can enter the cell by penetration, that's entry of the cell, by one of two means, either by endocytosis or by fusion of the virus envelope to the cell membrane. So those are the 2 ways an animal virus enters the cell. Now, let me mention, if anyone has an earlier edition of the textbook, that would be the first or second edition of the textbook made for Clark College, there's an error in the textbook, and it says that the virus can enter by pinocytosis. This is not correct. The virus enters the cell either by endocytosis or by fusion. Endocytosis is when the virus attaches to the cell membrane, and then the cell membrane invaginates around the virus, and that will become bigger, and then this will pinch off where the cell membrane will pinch together right there, and then this will pinch together right there, forming a vesicle, which will then move into the cell, bringing the virus into the cell. And that's endocytosis. Any question about that? If you've taken A and B, you know that when a ligand attaches to the cell membrane, the cell membrane frequently will undergo endocytosis, invaginating like that, and then separating into a vesicle, and the cell membrane will reform, and that will bring in the ligand into the cell. The same thing happens with the virus. The other thing that can happen, I don't think I have a picture of that. Not really, that's really endocytosis. So let me draw it in paint. So there's our cell. It's going to be a double membrane made of phospholipid. Well, the envelope is also a double membrane, largely made of phospholipids, by the way. When a virus attaches to a cell, and the virus has a envelope, that envelope can fuse to the cell membrane. See if I can do that right. Oh, that's pretty good. I'm not drawing that right. Let me figure this out. Yes, I am drawing it right. Let's go erase more of this one. All right, so the envelope fuses with the cell membrane, and now we got the cell membrane coming around the virus here. And let's make this a little virus in here. And as you can see, this virus now is now free to move into the cytoplasm because the cell membrane now becomes the envelope of the virus right there. And so the cell membrane now goes around the cell like that, and the virus is now inside the cell. Any question about any of that? And that's just because the envelope is a lipid bilayer and it can fuse to the cell membrane, which I tried to show you here. All right. Multiplication of animal viruses can happen by the lytic life cycle, where the host cell that becomes infected with the virus will replicate the virus. And then the last thing the virus does, it will lyse the cell, and that will release the newly replicated viruses. It's very similar to what happens in the lytic life cycle of a lytic bacteria virus. Same thing for a lytic animal virus, animal cell virus. But a virus can also follow the budding life cycle, and this does not usually kill the host cell. A budding virus is always enveloped. What will happen is the virus will replicate at low numbers, and that's a little different than a lytic virus because they usually replicate at high numbers, where the cell will make, I don't know, something like 50 to 200 viruses per cell, and then it'll lyse the cell, releasing all of those viruses. Well, the budding virus will have just a few viruses budding at one time. So this cell here is actually a cell, an electron microscopic image. I'm not seeing the entire cell here, but we can see 1, 2, 3, 4, 5, 6, 7, 8, maybe 9. And there could be a few more, but less than 9 viruses budding at about the same time. Now, they may not leave the cell all at the same time, but you can see that there's only a few viruses budding out of this cell at one time. And because the budding is not all at one time, and a lot at one time, the host cell usually can survive the budding life cycle. If the host cell dies, it's not from the virus budding out of the cell, it's from the host immune system recognizing that this cell is infected with a virus, and then the host cell can kill this cell to kill off the virus. Any question about any of that? So the budding life cycle does not usually kill the cell by the virus. And the virus itself can replicate at low numbers. So there we have the viral genome and the viral capsid protein. And then that virus starts pushing on the cell membrane or some other membrane of the cell. We'll talk a little bit about which membrane that can be. It will always begin pushing on a membrane that has viral proteins in it, meaning the genome codes for viral proteins and viral glycoproteins, which can be put into the, in this case, the cell membrane. And the virus will not bud here because there's no viral proteins there. It'll come up here where the viral proteins are and start budding here, forming a bud of the, in this case, the cell membrane. And it'll just rip that bud off. The cell membrane will reform, reanneal, and then this bud will also reanneal. And that is how the virus obtains its envelope, from ripping off a portion of the membrane of a cell, in this case, the cell membrane. So all budding viruses bud either out of a cell membrane or another membrane of the cell to form their envelope. And this can be the cell membrane, it could be the nuclear membrane, or it could be the membrane of the endoplasmic reticulum. It just depends on the viral species, which membrane it'll bud out of. I think most viruses bud out of the cell membrane, but a few, or some, bud out of the nuclear membrane, and a few bud out of the membrane of the endoplasmic reticulum. Any question about any of that? So those are the two main life cycles that animal viruses engage in, the budding life cycle or the lytic life cycle. Do realize there are other life cycles an animal virus can engage in. One of them is a permanent infection life cycle, which we'll talk about later. And the other is the, drawing a blank on the name of that one now. Let me move ahead and I'll find it because I'm not remembering the name of that. So we're on slide 32. Ah, I'm not finding it. Oh, there it is. It's a latent life cycle. Now I just remembered. All right, go back to slide 32. So the two virus life cycles that are minor are the latent life cycle and the persistent viral infection life cycle. And that makes all four of the viral life cycles. But the main two you need to know about are the budding and the lytic life cycle. Viruses have different genomes, and when they replicate, they'll have to follow a different replication cycle, depending on whether their virus genome is double-strand DNA, single-strand DNA, double-strand RNA, or single-strand RNA. So each virus type needs a slightly different mechanism of multiplication and biosynthesis to replicate its viral genome. Don't memorize the different family of viruses which cause human disease, but there are 7 families of DNA viruses that cause disease. Let me blow that up. Like I said, you don't need to know about them. We will talk a little bit about poxviridae, the pox viruses like smallpox, and herpes viridae. We'll talk a little bit about that. I don't think we talk about any of these others except for adenoviridae. We'll talk slightly about adenoviridae because this is one of the families of viruses that causes the human colds. And there are 3 families of viruses that cause human colds. And this is the 3rd most common, meaning the least common family of viruses that cause the human cold, adenoviridae. Herpes viruses, we've already talked a little bit about them, is a DNA virus. DNA viruses are generally replicated in the nucleus. The capsid proteins are made in the cytoplasm, so the gene will be transcribed in the nucleus, and then the messenger RNA will usually be processed and then sent in the cytoplasm, and then the proteins will be translated, making the viral protein codes and any other proteins that the virus needs. So herpes simplex virus is a DNA virus where the genome of the virus is DNA. Any questions about any of that? And then a rule of thumb, a DNA virus is generally replicated in the nucleus. Any question about any of that? There, have it there. Oh, there it is. There is one exception to the DNA viruses, and that is the pox viruses are a DNA virus, but they replicate in the cytoplasm. That's the only exception I know about for a DNA virus. All DNA viruses, animal cell viruses that replicate in the nucleus. Why? Because the nucleus has all of the enzymes and all of the cell parts needed for DNA replication. All right, once the viral DNA enters the host cell, one of 2 things will happen. First, the DNA of the virus can integrate into the host cell, not becoming a prophage because it's not a bacteriophage. This is an animal virus. So we use the term provirus. And that is when the viral DNA integrates into the host cell chromosome. If this happens, the provirus remains in the host DNA permanently. It is never excised. And then that host cell will permanently have the host, the viral DNA in it for as long as that host cell is alive. And whenever that DNA replicates, it will replicate the provirus. Any question about any of that? All right. The other thing that can happen is the host cell DNA can be expressed, new virions can be made. Obviously, we replicate the viral DNA. We also have to transcribe that DNA, make viral messenger RNA, make viral capsid proteins, which get together with the genomic virus to make the new progeny virions. And they will usually leave the cell either by the lytic life cycle or by the budding life cycle, because we're talking about animal viruses. So multiplication of the DNA virus in more detail. Let me blow this slide up. The virus can attach to the host cell membrane and then can enter the cell either by fusion of the virus envelope, if it has an envelope, to the cell membrane, or if it doesn't have an envelope or it doesn't fuse, the virus can enter the cell by endocytosis. The virus will be then released. The virus genome can be expressed, making viral messenger RNA. The virus genome can be replicated, making new genomic viral DNA. The new viral DNA can get together with the viral proteins, capsid proteins, to make the new virions, which will be released by the cell usually either by the budding life cycle, in which case the host cell generally remains alive and the virus can bud out, or it can be released by the lytic life cycle, in which case the cell is lysed, the cell dies, and that will be releasing the newly replicated virions, which are then outside the cell, which can then attach to another cell, starting the viral multiplication life cycle. Any question about this viral multiplication life cycle? All right, so that's how DNA family of viruses replicate. Remember, there are 7 families of DNA viruses that infect humans and cause disease. There are probably more that infect humans, but they don't cause disease, so they're not one of the 7 families. There are 11 or 12 I counted 15. 15 families of RNA viruses that infect humans and cause disease. We'll only talk about a few of them. You don't really need to know much about this table, so don't memorize it. We will talk a little bit about the family Picornaviridae. Not really about these. We will talk about the family Coronaviridae. We'll talk about the family Retroviridae. Paramyxoviridae, we'll talk just briefly about that. Orthomyxoviridae, we'll talk a little bit about that family, and all the other families we won't talk about at all. Talk about the type of DNA it is. If it is a single-strand RNA, there's the only double-stranded RNA. You need to state whether the strand is positive single-strand RNA or negative strand RNA. The positive strand can be read by the ribosomes. It'll have AUG on it. The negative strand cannot be read by the ribosome, and it does not have AUG on it. It would have the corollary to that, which would be UAC. Don't need to know about that. And there's the different diseases you can get from some of these families. Picornaviridae is the #1 family of viruses that cause the human colds. But Picornaviridae can also cause polio, hepatitis A, and you can notice that this family is very diverse. It's probably the most diverse family we'll talk about of animal viruses. Common cold is not a very serious disease. Polio is pretty serious. Hepatitis A is also somewhat serious of a disease. Coronaviridae is a family of viruses you are familiar with because COVID-19 is a coronaviridae, but actually coronaviridae was long known to humans long before COVID-19 came out, because this family of viruses is the second most common family of viruses that cause the human colds. Retroviridae are the viruses that encode the retroviruses like AIDS, feline leukemia virus. And this is the one family of RNA viruses that does not follow the rule of thumb. Generally, an RNA family virus replicates in the cytoplasm. The exception is the family Retroviridae, and it replicates in the nucleus, even though it is an RNA family of virus. It's the only RNA family that doesn't follow the rule of thumb that I know of. Any question about any of that? Paramyxoviridae causes many common childhood infections like mumps, measles, whooping cough. Orthomyxoviridae, you've probably never heard of that word, but the viruses which cause the flu are members of the Orthomyxoviridae family. Any question about any of that? Here we're looking at some RNA viruses, and you can see they're different shapes and sizes. The multiplication of RNA viruses is essentially the same with DNA viruses, but there are some differences because RNA is a genome instead of DNA. Viral RNA replication occurs in the cytoplasm. And when we're talking about single-strand RNA genome for a virus, you need to state whether it is a positive sense-strand RNA, single-strand RNA virus, or the negative sense-strand, antisense-strand RNA animal virus. The ribosome can read the positive single-strand RNA viral genome. It cannot read the negative single-strand RNA genome. Any question about any of that? All right. So for a DNA virus, you just separate the two strands of DNA, replicate each strand as a template, and then we have our viral DNA replicated. And then you transcribe the viral DNA to get the viral messenger RNA, which can then be translated into viral protein. For RNA, it's a little different. If it is double-strand RNA genome, you just separate the two strands of RNA, use one strand as a template to make the other strand. Only the positive single-strand RNA can be read by the ribosome to make viral messenger RNA, or be read as viral messenger RNA to make the viral protein. The negative strand RNA cannot be read by the ribosome. It does not make protein. If you have a positive single-strand RNA virus, it can be read by the ribosome to make viral protein. How you replicate positive single-strand RNA genome is you first make a negative single-strand RNA, and then you use this as a template to make more of the positive genomic single-strand RNA. Any question about any of that? If, on the other hand, your RNA genome is a negative single-strand RNA, you have to make positive single-strand RNA so that you can make the negative single-strand genomic RNA of the virus. And then that positive single-strand RNA can be read by the ribosome as messenger RNA to make the viral protein. Any question about any of that? All right, this is showing you negative RNA virus. Let me blow this slide up. And it will make positive single-strand RNA to use this strand as a template to make more genomic negative sense strand RNA. And now we got the negative sense genomic RNA made. You have to translate the positive sense RNA to make the capsid viral proteins, and the capsid viral protein will get together with the genomic RNA, the negative sense RNA, to make the new progeny virions. For positive single-strand RNA genome, you make the negative antisense RNA single-strand to use this as a template to make more of our genomic RNA, the positive single-strand RNA. And then you can read this positive sense RNA, or this one here, to make viral messenger RNA or use it as viral messenger RNA to translate that into the capsid viral proteins, which then can get together with the replicated positive single-strand RNA to make the new progeny virions. So positive sense strand RNA, the virus will come and attach to the host cell, enter the host cell either by pinocytosis or by fusion of the envelope, if the virus has an envelope, to the host cell membrane, bringing the virus into the cell. This is positive sense RNA. So to replicate it, you have to make negative sense RNA and then use that negative sense RNA to make more of the genomic RNA, which is the positive single-strand RNA. And then you can use the replicated positive single-strand RNA, either this one or the first one, to make viral messenger RNA, to make the viral capsid proteins. The viral capsid proteins will self-assemble into the virus, which will then package the viral genome into the virus, and then the viral genome will leave the cell either by the lytic life cycle or by the budding life cycle, depending on the species of virus, and that continues the life cycle of the virus. Any question about viral multiplication if it is a positive single-strand RNA virus? If it's a negative single-strand RNA virus, once again, the negative single-strand RNA virus has to attach to the host cell, enter the host cell by either endocytosis or by fusion of the cell membrane to the viral envelope, if the virus has an envelope. The viral genome will then be brought into the cell. The negative sense RNA will make positive single-strand RNA to use the positive sense single-strand RNA to make more of the negative genomic RNA of the virus. And then the positive sense strand RNA can be read by the ribosome to be translated into viral proteins. You largely make the viral capsid proteins, which will get together with the newly replicated negative strand RNA of the viral genome to make the new progeny virions, and they will leave the host cell either by the budding life cycle, which actually this sort of looks like that might be what's happening here, or it'll lyse the cell and the virus will be released by the lytic life cycle. Any questions about viral replication in the RNA family of viruses? If not, let's move on to the retroviruses. That's the family. Retroviridae. I do have time to talk about this, including lentiviruses, which you probably don't know of that term, but lentiviruses include HIV-1 and HIV-2, the viruses which cause AIDS, and also feline leukemia virus. sometimes called kitty AIDS. The difference is feline leukemia. The cat will eventually die of leukemia, but the leukemia weakens the cat's immune system, so the cat becomes sick with many other diseases. And I guess it could be the weakened immune system, which causes the death of the cat also. But the point is that both of these viruses, HIV and feline leukemia virus, are members of the family Retroviridae. Retroviruses are unique in that they contain the gene for the enzyme reverse transcriptase. Reverse transcriptase is a gene which first, see if I can find paint here. It first takes viral RNA which is, what happened to that thing? And it is positive strand, single strand RNA. And reverse transcriptase, I'm going to just abbreviate that as RT. I'm going to have to put it here because I'm too far up. And what it does is it makes single strand DNA from single strand RNA. And then the single strand DNA, see if I can copy this. Okay. Reverse transcriptase can work on single strand DNA to make double strand DNA. And that's how reverse transcriptase works, starting from oops, got it wrong. Starting from RNA and then making DNA, double strand DNA from it in 2 steps, but that's how it works. Any question about any of that? Well, for this then moves into the nucleus and it recombines with the chromosome. So this becomes integrated into the host cell chromosome. I'm not sure how I'll draw that. We'll just make the X chromosome. And it's going to go right here. And so that is right there, the provirus. Kind of. Provirus is what I'm trying to write there. Anyways, the double-strand DNA then will integrate into the host cell chromosome, and this will remain with the cell for the lifetime of this cell. And the only time the provirus will be replicated is when the host cell replicates its DNA, or when the gene is transcribed right here, meaning the provirus is transcribed. That can actually make viral messenger RNA.
I have a question.
Uh-huh.
So can the cell like not recognize that it's been invaded by a virus to like uh because in immune reactions, if like a eukaryotic cell gets
Uh-huh.
invaded by a virus, it like can break down whatever invades it and like signal to T helper cells that it needs to be like killed basically because it's infected so that it doesn't replicate anymore.
Uh-huh. All right. So if this provirus is not being expressed and the cell doesn't have any viral proteins or glycoproteins on the cell membrane of the cell, this cell cannot be recognized by the host immune system as being infected. And so this will continue. And this is one problem with HIV is it does integrate into your host cell chromosome. And even somebody who appears to be cured, you never know if at some point in the future, this uh, cell, which has the provirus in it, can activate, making viral RNA, which can then go out and infect other cells and cause the HIV infection again, causing the patient to eventually come down with AIDS. So we never know once this is integrated if the person is infected, because it might be only one cell in the patient, and yet if this virus activates, it could later down the road cause disease. But while it's not active, there's no way for the host cell to know this is infected, and then other than a human to know either, unless they were to happen to synthesize the DNA of the correct cell, they would not know that this was an infected cell also.
Okay, that makes sense.
Yeah. And even if you were to test somebody's like T cells, suppose you were to test, I don't know, 1,000 or even 1,000,000 T cells, but you didn't get the one that was infected. So even if the person appears to be uninfected, there could be one cell in the patient which has the integrated virus, HIV, in their cell. And sometime down the road, this could activate. It might not, but if it does, then the patient could start the HIV life cycle. More and more cells could be infected. And eventually the patient may, if they're not receiving medication, they could develop AIDS. But this is how this happens. And I guess I should continue this here. And that is viral RNA can be made. And this can then bud out of the cell. And then that can infect another cell. So we'll just say the life cycle continues. All right, any questions about retrovirus? This is the main point of showing you, and that is retroviruses makes double-strand DNA from viral RNA. It does it in 2 steps, first making single-strand DNA, and I don't think your book talks about that. Making first single-strand DNA and then making double-strand DNA from the reverse transcriptase acting on single-strand DNA. The reverse transcriptase protein is actually inside. There's one copy. I think it's right there. There's one copy of the protein inside the virus. This virus is a envelope virus. That's the envelope right there. These are the spikes, glycoprotein spikes. There's the capsid proteins. And it actually has, you don't need to know this, it's a positive single-strand RNA virus. There's actually two copies of positive single-strand RNA in the virus. You don't need to know that last part, but you should know it's a positive single-strand RNA. And what's really unique is it has reverse transcriptase. And I guess I shouldn't have shut that paint down, but you can see why this family replicates in the nucleus, and that's because it has a DNA intermediate in the replication life cycle. And that's why this has to replicate in the nucleus, even though this virus is an RNA family of virus. Any question about any of that? All right, for multiplication of retroviruses, attachment, penetration, and encoding happens like other animal viruses, but biosynthesis stage differs. We'll blow this slide up. So we have positive single-strand RNA, reverse transcriptase will make in two steps. This is only showing it in one step. First, it'll make single-strand DNA, and then the reverse transcriptase will use the single-strand DNA to make double-strand DNA. The double-strand DNA can be transcribed into positive single-strand RNA, which is both genomic, as well as the strand of RNA which can be read by the ribosomes to make capsid protein. The capsid proteins will get together with the newly replicated positive strand, single strand RNA to make the new progeny virions. And the new progeny virions will always bud out of the cell because this is a budding life cycle virus. And it will fuse with the cell membrane because it has an envelope. And then it will get inside the viral genome. The first thing it does is it will use the reverse transcriptase to change the viral genome in the cell. Let me put that over here. Well, can't get it over there, so I'll put it down there. To make the single-strand DNA and then use the reverse transcriptase to make double-strand DNA. The double-strand DNA can then integrate into the host cell chromosome, and that we call a provirus when the viral DNA integrates into the host cell chromosome. And let me state in a retrovirus that this viral DNA can integrate into different locations, different regions of the same chromosome, different chromosomes. And you can imagine that could muck up the cell. And you're getting lots of this virus in your body. It can integrate in different locations in different cells. And so a retrovirus does increase your risk of cancer, mainly because the viral DNA is just integrating almost at random. It's not quite random, but it's integrating at different locations in different cells. And so for our purposes, we'll just say it's integrating into the host chromosome at random. Once it's integrated into the host chromosome, the viral DNA can be transcribed to make viral messenger RNA. And that will be positive single strand RNA. That will be moved into the cytoplasm to make the viral DNA, especially the capsid proteins, which will then get together with the replicated viral RNA to make the new virions, which will always bud out of the cell at low numbers. And the low numbers is thought so that the losing of the cell membrane will not be critical to the cell, and then the cell's lifespan can continue. And why does the virus want to keep this cell alive? It's because it has a copy of its genome in it. Any questions about how retroviruses replicate? All right, that's enough
Dr.
about.
Volpe, I just wanted to let you know we're a few minutes past 6:20 now.
All right, let's stop here, and I'll talk about coronaviruses next time. All right, any questions? I will be in the lab to answer questions, but only for about 15 minutes. Once the last question is answered or the 15 minutes are up, I'll leave the lab. All right, any questions? All right, I'll see you in the lab in about 8 minutes.